Hello everyone, welcome to another tutorial. Today we'll try to analyze RNA-seq data from human sample. So basically, the Google Colab notebook today I'm showing you that this you can use for human and mice sample to analyze it with differential expression, and later on for visualization, like for producing heat map and uh, gene set enrichment analysis and cake pathway analysis. So anyway, first we need to extract the count data from the feature count output. For this, we can use this Google Colab. This link will be given in the description. First, we have to upload all the files, count matrix output file. These are we are uploading this to the Google Colab. Okay, all of my count data have been uploaded to the Google Colab. Now we have to convert it into CSV file. So basically, what we need to copy the path and paste it here, and now copy this expression number and paste it here. Here. Okay. Now run this code. This will produce this CSV file. You can see it here. So now, according to that, we have to convert all this text file into the CSV format. Okay, we have converted all the TXT file into the CSV format. Now we need to combine all this CSV file into a single CSV file. For that, we'll use this this code. So what will it actually detect this pattern in the name and combine all those CSV file into a single file. So basically, just run it. It will produce the CSV file, and it will be saved as combined raw count.csv. Okay, wait a bit, and it will be generated here. Okay, you can see that combined raw count CSV file has been generated. We need to download this file. This file will record for our downstream processing. Okay, open. This is the file. You can see that this user, these are the path of the file that has been uh, that this file has saved. So it is in the metadata of the file, so it came along with the column data. So we only need this much, this SRR code, this SRR exertion number for our files. Now what we need to we only need this SRR exertion number for our sample. So simply we can just remove from here, and yeah, this is fine. And you can see another thing, this is 78, this should be 79. Yes, 79, this 80, 81, 82, and 83. So this code actually arranges them according to our serial number or exertion number from lower to higher. Basically we can just do this, and this will be assigned to their exertion number automatically. Now we can use this data frame for our differential expression analysis and visualization. Okay, simply now we go to our second Google Colab notebook. This one, uh, in this notebook we'll use PyDESeq2 for differential expression analysis, scanp for volcano plot, and matplotlib and other tools for heat map and heat map production, and uh, PyGs and uh, G profiler for cake pathway analysis. So let's start. First, we need to install it. It will take some time. Okay, our package is been successfully installed. Now we need to load the package. Okay, now we need to load our data frame, count data frame here. So let's upload it first. The download this one. Okay, copy path and paste it here. Run it. Load. Our data frame has been loaded. Assign the gene ID as an index of this data frame. This code actually remove all the data which has zero in the all columns. So all rows have zero. Now we have to assign the metadata to this all these exertion number. Means what is this? Maybe this is control or treatment, whatever it is. We have to say this to the uh, Python, that this is this, and this is control, this is this treatment, this is this treatment, and treat them accordingly to that. Already I have loaded the metadata. You can change it according to your data. You have to change it according to your data. Okay. Okay, this metadata already for this data set only, so I just simply run it. Look, now all the metadata has been all the data has been assigned to their respective exertion number or respective column. Now differential expression analysis. Okay, our DDS data frame has been generated from all this data. Now we'll run differential expression analysis with PyDESeq2. It will take a bit of time. Okay, our differential expression analysis has been completed. Our DDS data frame gone through all this analysis, all this statistical analysis, and this produce this DDS data frame, which we can check it if it is good or not. If this is fine, this will return you with this type of uh, output. Look, objectives, condition, size factor, replaceable, and what kind of data is there. Normalized means non-zero, and you can say look on gene and okay, data frame is fine. Okay, uh, now we have to do statistical analysis to this analysis, like wand and all those things. Just simply run this, and here you again need to mention your metadata. What is your, which one is your control, which one is your treatment, all those things. According to your experiment you have to change it. Now statistical analysis. Okay, uh, now summary of this data frame. It will take some time. Okay, now see what we have. We have log to fold change, p value, condition versus this base mean, log ifsc value, stat, and all those things. All these statistical data we have for our data set. Now you can check. Basically in this we actually commanding uh, commanding are that this dataset to copy this dataset into the res dataset. So we just simply run res to call this dataset. Look, you can now we can see the data frame clearly. Look, first column is gene id, second column base mean, third column has log to fold change, fourth column has ifsc value, uh, and this is fifth column has static stat, and last two column has p value and p adjusted value. So these command actually saving this data set into this, that DESeq2 result treatment versus control. You can see that this data set has been generated already. You can download it and keep it here. Now we need sandbombings to annotate these data sets. If you are going to use mouse, you just need to write mouse here to get the mouse symbol or mouse annotation data. Okay, now you can see that respective to this exertion number, what gene it is, it's already been annotated. So we can use this dataset for our visualization and other purposes. This code actually remove all the data that in which has less than 10 base mean. This one, this column. Okay. Now for significant dataset, we need to use this one. It will sort the dataset according to p adjusted value, which is p adjusted value and log to fold change. Okay. Now you can see the dataset. Okay. This is our uh, significantly regulated dataset. Now for principal component analysis, we use scanp. So we need to install it and load the scanp. Okay. Our scanp has been installed into the Python Google Colab Python notebook. Now we use this dataset for principal component analysis. Okay, we just go through it and visualize it. Okay, you can see that this blue color means the control, and orange is SIEWS R1, and green is SIFL1 gene. This SI means uh, we can use microRNA to silence particular genes. So this is the if you see the SI one, is this means is the synthetic one that we use for transfecting the cell lines. Okay, now now for gene set enrichment analysis, we will use GACA gene set enrichment PY version that we will use for this analysis. We have to install it and load it into the Google Colab notebook. Now it has been installed. Now we have to ranking our data according to their statistics. Okay. So for gene set enrichment analysis, what it do, it actually take a gene set, a set of gene, and see that how much they have been enriched in that particular data set. It's been differentially, how much has been differentially expressed, and what is significant of those data set. So we have to give it manually. So for this, I am using PRC2 human gene set, this polycomb repressive complex 2 group proteins. So I have mentioned it which genes are actually involved in this pathway and this mechanism, and assigning them to the manual gene set. Okay, it will take some time. Now, okay, uh, it's done. So this is the data frame after After our analysis has been done, we can see FDR, the false detection rate, ES and NES value. So now we have to plot it. Okay, okay. This is our genset enrichment analysis figure. Now we will produce heat map. We are using NumPy and Cbond to execute this operation. Okay. Okay, this is a clustered heat map. Okay, this is the clustered heat map for particular gene set, which I have set it for PRC2 genes. Now for volcano plot. The two volcano. This is the global one. This is the local. G profiler for cake pathway analysis. Okay, when I am doing cake pathway analysis into this RES data frame that has been already designated. So this is our data frame after analysis has been done. Now we have to visualize it. Okay, now we can have this bubble plot of the cake pathway analysis. Okay, with this I finish my tutorial here. I hope hopefully this tutorial come handy into your research and learning how to do RNA-seq analysis for human and mice from human and mice sample. Wish all of you good luck. Thank you.